Going to talk about what happens in the proximal convoluted tubule, the PCT, which is this first region of the nephron after filtration has occurred. And this is where about 67% of all our filtrate is reabsorbed. Almost 100%, so 99 or so of the nutrients, so glucose, amino acids, etc. So a lot of reabsorption occurs, right? We're going to have a little bit of secretion as well. So in order for reabsorption to occur, we've got to pass through this apical surface and the basolateral surface of this simple cuboidal epithelium, and then into, right, our paratubular capillaries. This is going to rely on the mechanisms we saw in the previous video related to transepithelial transport, and hopefully ones that you remember from before. So first I want to go through what those things are we'll be talking about, and then we'll draw them, how they do it. So first of all, we're going to have sodium. We've got to get it through the apical surface. That's going to occur through some leak channels, some co-transport, some antiport. So both co-transport and antiport are moving along with or the opposite way of another molecule. So for example, sodium glucose co-transport. On the basolateral surface, we're going to have active transport, our sodium potassium pump. There's a lot of ways to move sodium. It's really important to reabsorb. So we'll look at that. I already mentioned glucose. So glucose, amino acids, vitamins and other nutrients, some other ions are going to move through this apical side via secondary active transport, co-transport with sodium. That's going to allow them to get into the epithelial cell. Then we're going to have also facilitated diffusion. And what determines which one of these occurs where is where the gradients are. So electrochemical gradients. All right. We have water we want to reabsorb. It's going to follow the solutes. This is osmosis. So it's going to move through the plasma membrane, through the phospholipid bilayer itself, as well as aquaporin, the water channels that facilitate water movement, and it's going to follow the way the solids are moving. So this is osmosis. We've got other ions, so chloride, calcium, urea is not an ion, but also similar in that it's going to use paracellular, so go between the cells primarily. And this is going to be using some passive diffusion, so using electrochemical gradients. Lipids, similarly, are able to use passive diffusion, but they go actually through the plasma membrane. So transcellular. And lastly, we've got bicarbonate, an important buffer that we want to be able to reabsorb. This is going to be linked to hydrogen secretion. So this is one thing that is secreted, and sodium reabsorption. So these things are all things that are reabsorbed besides this hydrogen secretion. So let's make sure we can see all these processes on this cross section here. Again, here's our lumen, zoomed in right here, apical, basolateral, capillary. So we've got to move sodium and potassium. Sodium is going to go out here, potassium in. We'll talk about potassium secretion in the distal convoluted tubule. So because of this gradient, we can move glucose and sodium in. So these that is actually reabsorption of glucose and sodium glucose cotransporter. Once glucose is inside the cell, it can diffuse out through the glucose transporter. This is facilitated diffusion. Amino acids and some vitamins, some other ions are going to be the same mechanism as glucose. We've also got sodium moving in through some leak channels. So this is a leak channel. Leak channels could exist for various ions. This is a leak channel for sodium because sodium is moving through it. I've got another leak channel shown down here. This is actually going to be for hydrogen secretion. So I'm going to draw secretion in red because that's what I have for my key here. So this is hydrogen ion going this way. I'll come back to that in a moment. Actually, I'll come back to that now. That hydrogen is going to, we want to secrete it. It's actually going to be secreted via antiport. What do you think could antiport hydrogen? How could we be something that is going to go down its gradient and be a positively charged thing to be electrically neutral? Sodium. So here's another way of bringing sodium in. And these are all reabsorption, which my key I have blue. Reabsorption this way. So look at all these mechanisms for sodium. It needs to be reabsorbed. That's really important for maintaining osmolarity of our blood. It also is going to be helpful for us bringing glucose, amino acids in, and secreting hydrogen out into the urine, that acidic hydrogen ion. So that blue, the purple thing I have right there, that is our antiporter, specifically sodium hydrogen antiporter. So we've got everything being reabsorbed besides hydrogen that we've seen so far. So following all that movement of the ions is going to be water. Water moves in through aquaporins. Then we've got lipids can just diffuse. Some other substances such as chloride, some other ions are going to actually diffuse in between the cells. They can sneak in there. Okay, that's most of it. One more thing though. Bicarbonate. We want to be able to reclaim bicarbonate. And that's going to be linked to this hydrogen ion secretion. So that hydrogen ion is coming in from the blood, we're secreting it. That's actually passive diffusion. That hydrogen ion is going to combine with bicarbonate. I'm sorry, it's not, no, it's not. That hydrogen ion, I already had it, it's going to flow out that way. For reclaiming bicarbonate, we are talking about what's in here, right? So I'm going to draw you this reaction, what's happening in the lumen. Should look familiar. We've got hydrogen ions, we've got bicarbonate. Those can form water and carbon dioxide. Why would we want to do this? Because both of these things can pass easily through this membrane. Bicarbonate is negatively charged, hydrogen ions, there's no drive for it. We actually don't want it to go this way, but it's the bicarbonate that we can't easily get across this membrane, whereas water and carbon dioxide can. So those two things are going to then diffuse either through facilitated diffusion, or carbon dioxide can just pass right through so simple diffusion. They're going to go in this way. Inside the cell, Are we just going to have them diffuse out? No, because we want to reclaim bicarbonate, but not hydrogen ions. So we're going to have the reaction occur in reverse. I'm sorry, that's the wrong thing. H2O plus carbon dioxide, that's what just diffused in. Those are going to form our hydrogen ion and H and bicarbonate. Now we can have bicarbonate be reabsorbed. It actually moves along with sodium as well. Imagine that. Plus sodium. And our hydrogen ion, what's going to happen to it? It's going, that's it right there. It can go out that way. So it's a way of reabsorbing bicarbonate, which is that buffer, while secreting hydrogen ion.